So on this video we are going to be looking at multiple alleles and the genetics behind multiple alleles and how we would approach solving multiple allele problems. So as a reminder, individuals carry two alleles for each autosomal gene. The alleles, you get one from mom, one from dad, and remember an autosomal gene is a gene that controls some trait about you except whether you're male or female. So our autosomal genes give us every trait about us except being a boy or girl. So a gene can have multiple alleles because its sequence can deviate in many ways. So there's different allele combinations that would produce a variation in phenotypes. An example is PKU gene. It has hundreds of alleles that result in 4 different phenotypes. Now, incomplete dominance, the heterozygous phenotype is different from the two homozygous. The example you would typically see in a biology class is big R is red with plants. Little r would be white flowers in plants, and big R, little r would be pink flowers. In humans, an example would be familial hypocholesterolemia, and that's abbreviated FH. The heterozygous has approximately half the normal number of receptors in the liver for the LDL, which is the bad cholesterol. And so a homozygous individual for the mutant allele lacks the receptors, and so their cholesterol level is going to be extremely high. However, the prime example that's used with multiple alleles typically is blood type. In the 1900s, Karl Landsteiner observed that there were 4 different types of blood. There was type A blood, there was type B blood, there was type AB blood, and there was type O blood. And that was due to the antigen-antibody reactions. Now remember that a person that has type A blood, they still received 2 traits for the blood type. They received one from mom and one from dad. So a person that is B blood, they received a trait from mom for blood type and a trait from dad for blood type. So if we look at type A blood, they can be AA, meaning they received the A from both parents, or they can be AO. They received an A from one parent and an O from another parent. A person that's type B blood can be BB, meaning they received a B trait from each parent, or they can be BO. They received a B from one parent and an O from the other parent. A person that's type AB blood receives an A from one parent and a B from the other parent. A person that's type O receives an O from both parents. Now, about 45% of the population is type O blood. About 42% of the population is type A blood. Right around 12% of the population is B blood, and around 4% of the population, roughly, is AB. That may not quite add up to 100, but it's close, and that's not counting the RH factor. O is the universal donor, and AB is the universal receiver, meaning blood can be given to any individual, and AB can receive any individual's blood. Now, when we're looking at the antibodies on here, this is what they will react against. So a type A blood, on the blood cells, the outside of the blood cells, they have a reaction where they will react against B. And so they cannot receive any blood type that has a B in it. So an A individual could receive A blood, they could receive O blood. So they recognize A as being acceptable. O is not recognized one way or the other. B is recognized as being something that they would react against. So these are the antibodies, these are the anti-antibodies, or the antigens, which are the friends. A B person has A antibodies, would react against any type of blood that has A in it, so they could not receive A blood, they could not receive AB blood. They could receive B, they could receive O. An AB does not have any antibodies. It will not react against anything. and that's why it can receive all blood types. So it can receive A blood, B blood, AB blood, and O blood. O blood has antibodies for both A and B, so it reacts against both, meaning O blood can only receive O blood, and that's why O blood is the most common blood type. You do need to know this chart, so I would copy this down. The blood type is also called codominance. Besides being called multiple allele, it's also called codominance. This right here would be your AA blood. This would be your AO. Here you have BB, BO. You have your AB blood, and then your O blood. I don't particularly like this way of writing, and that's why to make things easier on you, you should copy down the chart that was a couple of slides back. That will be much easier for you than using these. However, if you were to get a degree in genetics, this is the typical key that is used in upper level genetics classes for blood typing. And here we just have a few Punnett squares showing you how they work out. Type A blood, where you have AA and then B, bring it down, bring it over, then you would have all being type AB blood. Here if you had an AO crossed with a BB, then you can have AB and BO as the blood types, and so you can just look through these Punnett squares. So there is a unusual condition that can exist in blood type. This is a epistatic condition where we have another gene affects the blood type that's expressed. And so it's the H gene is epistatic to the I gene, which gives the blood type. And the H protein is on the cell surface just like the A or the B antigens are on the cell surface of the blood cells. So if a person is little h on their genotype, they don't have the H protein. And without that H protein, then they can't recognize the A or the B antigens. So an individual who has a genotype of little h, when they do their blood test, phenotypically, they're going to test out as being type O blood. However, if you were to look at their blood in more detail, their blood type could be A blood, B blood, AB, or O. It could be any of those. And there have been situations where a woman had her child taken away from her because the child had a similar appearance to a child that was missing. Police pull them over. She goes, "No, that's my child." This is back before they had the DNA test. They did blood test, and mom and the child didn't match up. And they didn't match up. Mom was matching up as type O blood, and the child was matching up as type AB. And that's not possible. But what happened was mom had that little h genotype that was causing her to phenotypically test as type O blood, even though she was another blood type. And so the child was actually hers, but because she had this condition, blood type would not prove that. So a little bit more about the Bombay phenotype. It was discovered in 1952. A woman in Bombay was showing a kind of an unusual genetic history with her blood type. She was in need of a transfusion. They found out that she lacked both the A and the B antigens, so they assumed that she was type O. However, it was shown that one of her parents was type AB blood, and she had donated a B allele to two of her offsprings. So looking at this woman, she should be genetically type B blood, but she was showing up functionally as being type O blood. And that's where this epistasis of the H protein came in, and they discovered this condition. So around 1930, 1940s, the Rh antigen was identified. It was first identified in rhesus monkeys. And here we have a rhesus monkey right here. And then later was identified in humans. Now, Rh positive is dominant, so an Rh positive person can be positive positive or positive negative. Rh negative must be negative negative. 85% of the population is Rh positive, and about 15% of the population is Rh negative. So the Rh antigen really only plays a factor if you're getting a blood transfusion or when a woman is pregnant. And so if a woman is pregnant and she is Rh negative, she will need to receive either gamma globulin or RhoGAM shots during pregnancy or within 48 hours after giving birth, or she will build up antibodies against her baby. And what that means is with her next pregnancy, her body will see the developing embryo as being a reoccurrence of the infection and will attempt to attack the fetus. And what it does is it attacks the blood cells of the individual and it tries to kill the red blood cells. So we call that disorder erythroblastic fetalis. There are also other ways that we can look at the blood. I mean, we have the A, the B, the AB, the O. We also have the Rh factor, and then we have the MN blood group. And the MN blood group, an individual who inherits two N's would be NN. A person that has two M's would be MM. And a person that's heterozygous would be MN. And so when an individual is getting a blood transfusion, they want to match up the blood as close as possible. And when a person is having their DNA tested for paternity, if they're looking at the blood, they would have to look at, okay, let's look at their ABO type. Let's look at their Rh factor. Are they positive or negative? Let's look at their MN factor. And so there's quite a few genes associated with the blood type. So a couple of other genetic traits associated with blood are the S antigen. Now the S antigen is very closely related to the MN. So an S person can be SS, or they can be big S little s. They can be little s. So I kind of got this. This is how we would write that down, showing this individual is MN, big S, little s, crossed with NN, big S. And so you would have to work this out like a dihybrid problem. And then some individuals are secretors, meaning that you can tell their blood type through body secretions like the eyes or the nose. About 78% of the population are secretors. To be a secretor, you can be big H or big H little h, and then to be a non-secretor, you are little h. So in rabbit coat color, rabbits, the wild type is called Algati, and what that means is that there's bands of hair. So it'll start off gray, and then it goes yellow, and then the tip is black. Albino means they're completely white because they're lacking pigment. Then there's chinchilla, which is a black-gray color, and then there's Himalayan, which is white with black extremities. Okay, so on height, we could say that big T is tall, and so an individual could be big T and be tall, or they could be big T, little t, and be tall. And then we see that tall is dominant over short. The only way to be short would be little t. Well, I do want you to pause, and I want you to write down this key right here. You will need it for your homework and for some of your tests. On this right here, rabbit coat color, there are 4 colors: Algati, chinchilla, Himalayan, and albino. Algati is dominant over all of these. So genotype, they could receive an Algati from both parents. They could receive an Algati from one parent, chinchilla from one parent. That would still show up as being Algati. They could receive an Algati from a parent and a Himalayan from a parent. They could receive an Algati from a parent and an albino from a parent. Again, Algati is dominant to all of these others. Chinchilla, they could receive the chinchilla trait from both parents. they could receive a chinchilla trait from one parent and Himalayan from the other, a chinchilla from one, and albino from the other. So chinchilla is dominant to Himalayan and albino. Himalayan, they can receive the Himalayan trait from both parents, or a Himalayan trait from one parent and albino from the other. So Himalayan is dominant to albino. Albino, the only way to be albino is to receive the albino trait from both parents. Do write this down so you have this for your homework. And so this makes a little bit more sense to you. This right here is my Algati rabbit here. This is what a chinchilla looks like. This one right here is a Himalayan, and then this one right here is an albino.